Hello, everyone. Welcome to lecture number 4, part 1 of our medical biochemistry series on the chemistry of carbohydrates. I am Dr. Muzamil Liaqat Ali, assistant professor of biochemistry. In the previous lectures, we studied monosaccharides, their cyclic structures, anomers, meter rotation, sugar derivatives, and chemical properties. Today, in part 1, we will focus on the formation of glycosides, glycosidic bonds, and the structure and properties of important disaccharides. These concepts are foundational for understanding how sugars link together to form complex carbohydrates and how they function in drug and nutrition. So let's begin. Formation of glycosides. Glycosides are formed by condensation between the hydroxyl group of the anomeric carbon of the monosaccharide and a second compound, which may be another monosaccharide or a non-sugar molecule called as a glycone. When linked together, OH group and O-glycosidic bond is formed, resulting in a glycoside to form or from the glucose or the glycoside from the galactose. When linked to an amino group, an N-glycosidic bond is formed. For example, between adenine and ribose in nucleotides such as ATP. This reaction is the chemical basis for building all oligosaccharides, polysaccharides, glycoproteins, and many important drugs. So, what is its medical importance? So, let's discuss in detail. Many glycosides have significant therapeutic value. Cardiac glycosides such as digoxin and orban contain a steroid as a glycan and act by inhibiting the sodium-potassium ATPase pump on cardiac cell membrane. They are used in the treatment of heart failure and arrhythmias. Other medically important glycosides include the antibiotic streptomycin. The sugar portion often improves the drug solubility and specificity. The glycosidic bond is created when the anomeric hydroxyl group of one sugar reacts with hydroxyl or the other suitable group of another molecule, releasing a molecule of water. General reaction, as you can see on your screens, right? The R group or the OH, the anomeric, that is anomeric, plus the HO or reacts with to form a different compound by releasing a water molecule. For example, methanol reacting with glucose forms methyl glucoside. The bond can be alpha or beta depending on the configuration of the anomeric carbon. These bonds are stable in the body but can be specifically hydrolyzed by enzymes during digestion. Now, let's discuss the amino sugar, the hexosamine in complex molecules. Amino sugars such as D-glucosamine, D-galactosamine, or that is also known as chondrosamine, and the D-menosamine are essential components of glycoproteins, gangliosides, and glucosamine glycans. For example, the hyaluronic acid and the chondroitin sulfate. Several antibiotics, including erythromycin, contain amino sugars that are critical for their antibacterial activities. Let's have a general overview of the disaccharides. Disaccharides consist of two monosaccharides units joined by a glycosidic bond. Glucose is almost always one of the units. The second may be the fructose, the galactose, or another glucose. Disaccharides are formed by condensation, the removal of water, and broken down by hydrolysis, that is the addition of water during the digestion process. Right? The three most important disaccharides in human nutrition are the maltose, sucrose, and lactose. Now, let's discuss the maltose. Maltose, the malt sugar, is composed of two glucose molecules linked by an alpha 1,4 glycosidic bond. It is produced during the partial hydrolysis of starch by salivary or pancreatic amylase. Maltose is also used as a nutrient, sweetener, and in fermentation. It is the reducing sugar because one glucose unit retains a free anomeric carbon. Next is the cellobiose, right? It is basically the beta-1,4 linked disaccharide obtained from cellulose. It differs from the maltose only in the configuration of the glycosidic bond, that is beta instead of alpha. So humans cannot digest cellobiose because we lack cellulase, where maltose is readily hydrolyzed by maltase. This highlights how the stereochemistry of a glycosidic bond determines digestibility. Now, the sucrose, the table sugar. Sucrose consists of alpha D glucose linked by beta D fructose by an alpha 1,2 glycosidic bond. It is obtained from the sugarcane and sugar beet. Sucrose is a non-reducing sugar because both anomeric carbons are involved in the linkage, leaving no free aldehyde or ketone group. This is why sucrose does not react with Benedict reagent unless first hydrolyzed. Now, the lactose. Lactose, the milk sugar, is made of beta D-galactose joined to alpha D-glucose via a beta-1,4 glycosidic linkage. It is the main carbohydrate in the milk and known as milk sugar. Milk contains both alpha and beta anomers of the lactose, right? Lactose is a reducing sugar because the glucose unit has a free anomeric carbon. It is used in infant formulas and pharmaceutical preparations. Next is the trehalose. Trehalose is a non-reducing disaccharide in which two alpha D-glucose units are linked by an alpha 1-to-1 glycosidic bond. It is naturally present in mushrooms, insects, fungi, and bacteria. In the food industry, trehalose is used as a texturizer, stabilizer, and a humectant due to its low sweetness and excellent stabilizing properties. So now let's discuss the sugars and artificial sweeteners. Different sugars vary in sweetness. Sucrose is taken as the reference standard with the value of 100. Fructose is sweetener than sucrose, while lactose is much less sweet. Understanding this or these differences is important when advising patient on diet, especially in diabetic and metabolic disorders. So we have now completed the part 1 of our lecture number 4, the chemistry of carbohydrates, covering glycosidic bond formation, clinically important glycosides, and the detailed structure and properties of the major disaccharides, maltose, sugar, lactose, and trehalose. And in part 2, we will discuss the carbohydrate digestion and the gastrointestinal tract and the clinically very important topic of lactose intolerance. Thank you very much for watching the lecture number 4, part 1. If this part helped you understand glycosidic linkages and disaccharides clearly, please like the video, share it with your fellow medical students, and subscribe to the channel for the complete high-yield biochemistry series. Hit the notification bell so you don't miss the part 2. So drop your question about glycosidic bonds and disaccharides in the comment section. I'll reply to them right. So this is Dr. Muzamil Liaqat Ali signing off from the part 1. Keep revising the alpha and beta linkages daily. See you in the lecture 4, part 2. Thank you, and keep learning biochemistry with a passion.